Hello, everyone, and welcome to today's live broadcast. Is free 25OH vitamin D the next generation vitamin D marker? Presented by Nicolas Heureux. He is a principal scientist at DIAsource Immunoassays. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Nicolas Heureux. I will now turn the presentation over to him.
Good morning or good afternoon, everybody. My name is Nicolas Heureux. I'm based in Belgium. My background is in organic chemistry, and I work for DIAsource Immunoassays for 7 years now, starting in the development of vitamin D assays and kind of working the area of free vitamin D, which will be the topic of our today's webinar. And the title of the webinar is free 25-hydroxy vitamin D the next generation vitamin D marker? This is the outline of the presentation. I'm going to start with an introduction on vitamin D metabolism. It will be a very general introduction, but it will allow us to really set the ground for the rest of the presentation. Then we're going to move to total 25-hydroxy vitamin D and then introduce the free 25-hydroxy vitamin D concept. I'm going to then discuss the different conditions which affect the free and total equilibrium, and then present you a few key studies on free 25-hydroxy vitamin D. At the end of the presentation, I will explain you how you can measure or evaluate the concentration of free 25-hydroxy vitamin D, and then there will be some take-home messages and a Q&A session. So let's start with the vitamin D metabolism. And as you probably know, vitamin D is sourced from two different ways, or two different sources. The main source of vitamin D comes from the sun. And the sun is transforming the 7-dehydrocholesterol by the action of UVB into vitamin D3, which is drawn on the slides. The other source of vitamin D is food or supplements, and the food can be fortified with vitamin D or not. Food is usually very poor in vitamin D. Only some food sources like fat fishes contain significant vitamin D. From food or from supplements, you can have two different forms of vitamin D, the D3 and the D2 form. Once the vitamin D is in the body, it is transported to the liver, where it is transformed by an enzyme into 25-hydroxy vitamin D. 25-hydroxy vitamin D is then further converted in the kidney by the 1-hydroxylase enzyme into 1,25-dihydroxy vitamin D. Other tissues or organs are also able to do this transformation, and so to obtain 1,25-dihydroxy vitamin D for 25-hydroxy vitamin D. The 1,25-dihydroxy vitamin D is then excreted through 24-hydroxylation, so it goes to 24,25-dihydroxy vitamin D or and 1,24,25 OH3 vitamin D. 25-hydroxyvitamin D is currently the best marker of vitamin D deficiency and is used worldwide in all laboratories and hospitals to really evaluate if someone is vitamin D deficient. People sometimes are going to talk about vitamin D or 25-hydroxyvitamin D or total vitamin D. All these terms, usually they refer to 25-hydroxyvitamin D. What we mean by total 25-hydroxyvitamin D is two different things. Some people will mean total is the sum of the D2 and D3 forms. And as you will see later in this presentation, total can also mean the free fraction plus the bound fraction. So if you use the term total, it can refer to two different concepts. Now I'm going to talk a little bit about total 25-hydroxyvitamin D, which, as I said before, is currently the best marker to evaluate vitamin D deficiency. It is used by laboratories and hospitals in different situations, and the first one, of course, is to diagnose people for vitamin D deficiency. So are you vitamin D levels too low? In that case, you can be classified as vitamin D insufficient or deficient, depending on the concentration. So this is really the first application of this test. The second application is to monitor people which are supplemented with vitamin D. So after you have diagnosed people for vitamin D deficiency, you will provide them with supplements, and the vitamin D test is also there to monitor the treatment. So to see if the supplements amount is sufficient to reach optimal levels of vitamin D. The last application of this test is a little bit less of use, but can be used to diagnose vitamin D toxicity in case of huge intakes, but that's a very rare situation, and it happens mainly because of manufacturers' mistake when they manufacture supplements. You can see on those two graphs that the number of vitamin D tests in laboratories has really increased a lot the last 5 years. Per example is from a laboratory in Calgary area, and you really see that the number of tests has really exploded the last years. The graph below, I think, is from France, also showing an increase in the number of vitamin D testings. There are different reasons for that. People have been discovering new applications. People awareness also increased. The number of assays on the market, especially automated assays, has also increased. All these things explain the situation why we have so many vitamin D tests now running in the laboratories. There are some issues associated with this 25-hydroxyvitamin D tests. The first one, and a lot of people are talking about that, is laboratory-to-laboratory variability. So if you test the same sample in different laboratories, you might find discrepancies or different results. And for that, the VDSP program has been created. VDSP stands for Vitamin D Standardization Program. And the aim of this program, the objective of this program, is really to standardize vitamin D assays and try to reduce those discrepancies. The next issue with 25-hydroxyvitamin D concentrations is the inconsistency in reference values. On this table, which I'm sorry for that is in French, you can see that different institutions from different countries and different areas of the world have established different reference values. So in some countries, you might speak about deficiency below, for example, 10 nanograms per ml. In some other countries, it will be below 20, etc., etc. And that's a main concern, and people do not really know which one they have to use, depending on which institution they rely. The third issue with 25-hydroxyvitamin D is the inconsistency in some clinical studies. I see quite a number of clinical studies which are published and which show opposite results to each other. So some clinical studies will show a benefit of vitamin D versus a certain disease. Some other clinical studies will show a negative effect of vitamin D against the same disease. And if you do meta-analysis of all these studies, you end up with a graph like it is presented here, with effects which are positive or negative. So at the end, people do not really know what to think. What are the reasons for those issues? Well, for calibration issues and for laboratory variability, there is obviously a calibration issue. And for a long time, there was no reference method or no reference sample for vitamin D. The VDSP program has really helped there to establish reference methods, to establish reference materials, and to discuss with manufacturers and laboratories to try to harmonize vitamin D assays. And those calibration issues now more or less disappeared from the laboratory. So this is now a much better situation. For the two other points, so the inconsistency in reference values, but also in the results of clinical studies, there is no easy explanation. One of the explanations might be that actually total 25-hydroxy vitamin D might not always be the optimal marker. And you can see now that there are some other vitamin D markers that are used in some cases or not. And the first one is 1,25-dihydroxy vitamin D, the active form of vitamin D. This one is used in laboratories since probably more than 20 years, but is very specific for one application and is usually tested when you have a problem with your kidneys, so when you have kidney disease. It is not a marker of vitamin D deficiency. 24,25-dihydroxy vitamin D is a recent hype, let's say. It has been studied a little bit those last years and is now used or can be used in the diagnosis of idiopathic infantile hypercalcemia, so IIH. The C3 epimer of 25-hydroxy vitamin D has been also studied a little bit. So far, people have not found a clinical utility for it, but it has an importance in the accuracy of the measurement of 25-hydroxy vitamin D because it can interact with the results you obtain in your vitamin D tests. The non-hydroxylated vitamin D form, so the form you really source from the sun and from the food, has also received a little bit of research, but it's not yet very clear what is the role of these metabolites. And the last one is free 25-hydroxy vitamin D, which is obviously the topic of this webinar and which I'm going to discuss in the next slides. Let's talk about the concept of free 25-hydroxy vitamin D. And let's start with this slide. So on this slide, you see how 25-hydroxy vitamin D is distributed in the body. 25-hydroxy vitamin D is a hydrophobic molecule, so it is really not soluble in water, so not soluble in blood or plasma. So it is transported by binding proteins, like a lot of small molecules. And in the case of 25-hydroxy vitamin D, the main transport protein is DBP, vitamin D binding protein, which binds about 88–90% of 25-hydroxy vitamin D. Albumin, which is a more general protein, can also bind about 10–12% of 25-hydroxy vitamin D. Although the concentration of albumin is much higher than the concentration of DBP, the affinity of albumin for 25-hydroxy vitamin D is much lower. So this is the reason why it only binds at 10–12%. And then on the bottom of the slide, there is a small fraction, which is the free form, so the form that is not bound to any protein. This small fraction represents about 0.04%. So it's a very, very small fraction, but you will see it is very important. So when we talk about total 25-hydroxy vitamin D, which is the concentration, the test that people are running at the moment, it is the sum of these three fractions, the DBP fraction, the albumin fraction, and the free fraction. Typically, the concentration are between 10 and 50 nanograms per ml. Free 25-hydroxy vitamin D is this red fraction. It's a very small percentage of the total, and in terms of concentration, we can talk about 2 to 8 picograms per milliliter. Let me introduce you now the free hormone hypothesis. This hypothesis says that the biological activity of a given hormone is affected by its unbound or free, rather than protein-bound concentration in the plasma. And you have examples existing for years in papers, in literature, and in laboratories as well, like the free thyroid hormones, so free T3 and free T4, which are now measured a lot, and sometimes even more than the total concentrations. Free testosterone and free cortisol are two other cases. So in those cases, people have found that the concentration of the free hormone is better linked to its biological activity than the concentration of the total hormone. And this was proven in research, but also in IVD laboratories. Now, for vitamin D, the situation is slightly different, and I'm going to explain why. 25-hydroxyvitamin D, which is the parameter that laboratories are measuring, is not the biologically active form of vitamin D. It is 1,25-dihydroxyvitamin D. So on one hand, we have a biologically active form, the 125, but on the other hand, we are not measuring this form. So is the free hormone hypothesis also valid for vitamin D? That's the question. On this slide and on this picture, you see that 125-dihydroxyvitamin D is formed into some cells, for example, in that case, proximal tubule epithelial cells. And 125-dihydroxyvitamin D is formed in those cells from 25-hydroxyvitamin D. There is a conversion inside the cell. For 25-hydroxyvitamin D to be transported into the cells, there are two options. Option A is a diffusion through the membrane of the free form. Because the form that is bound to binding proteins is too big, is too hydrophobic, it cannot cross the cell membrane. So in mechanism A, the free form of 25-hydroxyvitamin D crosses the membrane and is then converted into 125-dihydroxyvitamin D, which is the active form. In mechanism B, bound 25-hydroxyvitamin D, and mainly the DBP bound form, is internalized through a protein receptor named megalin, and once it is internalized, it can be liberated from the binding protein into 25-hydroxyvitamin D, which is then converted into the 125-dihydroxy form. So there are two mechanisms possible, and this was, of course, studied by different people, and people found that in most extrarenal tissues there is no megalin, so the passive diffusion mechanism is the mechanism in those conditions. In that case, what is important is the concentration of free 25-hydroxyvitamin D. Because the concentration of free 25-hydroxyvitamin D outside of the cell is linked to the concentration of 25-hydroxyvitamin D inside the cell and is there linked to the concentration of the active form. In renal tissues, the situation is different. There is megalin uptake, and in that case, the two mechanisms are present, with the megalin mechanisms being the majority. In that case, the total 25-hydroxyvitamin D concentration is the most important, and not anymore the concentration of free vitamin D. I'm going to show you now how the free total vitamin D equilibrium is affected by different conditions. What we have seen from the slide just before is that the concentration of free 25-hydroxyvitamin D is at least as important as the concentration of total 25-hydroxyvitamin D. If you take a healthy monoethnic population, the free 25-hydroxy concentration is always the same percentage of total 25-hydroxyvitamin D. This percentage is represented by the letter Y here. So it means that, let's say it's, for example, always 0.03% of the concentration of total vitamin D. In that case, even if free vitamin D is important, you can just measure total vitamin D as is done now in laboratories. And from that point, you can have a good evaluation of vitamin D deficiency. Because it will be translated automatically into the concentrations of free vitamin D. If you are deficient measuring total vitamin D, you will also be deficient measuring free vitamin D. There will be no difference in the evaluation of vitamin D deficiency. In other conditions, the situation is totally different. And the first conditions are the ones in which the concentration of the binding protein DBP is altered, is modified. And those conditions are listed here. It's renal disease, pregnancy, liver disease, intensive care patients, women taking oral contraceptives or hormone replacement therapy, obesity, and proteinuria. Let's take the example of pregnancy. During pregnancy, and that's seen from the first trimester to the third one, DBP concentration increased by about 50%. In that case, because you have more binding protein, you will have more 25-hydroxyvitamin D in the bond form, and you will have less 25-hydroxyvitamin D in the free form. Because of that, you will also have less 25-hydroxyvitamin D inside of the cell, so you will have less of the active form of vitamin D. So if you measure total 25-hydroxyvitamin D in pregnant women, you will measure mainly the bond form. And actually this does not represent anymore the biological action of vitamin D, because in that case the biological action is driven by the concentration of free 25-hydroxyvitamin D, which is reduced in this case. The risk for this population is that you will misclassify people when measuring total vitamin D, total 25-hydroxyvitamin D. You may think that a pregnant woman has optimal levels of vitamin D because you are measuring the total. Actually, she might still be deficient because what is important in this population is to measure the concentration of free vitamin D. So that's the first condition, which is pretty important. The other condition, or the other situation in which the concentration of free vitamin D is very important is when you work with different ethnicities. The reason is that those different ethnicities will have different forms of vitamin D binding protein. These are polymorphs, and there are in total 6 different polymorphs for DBP. For example, blacks and Asians have mainly the GC1F form, and whites have mainly the GC1S form. Okay, why is this important? Because those different forms have different affinities for 25-hydroxyvitamin D. For example, in blacks and Asians, where the GC1F form is the major form, you have a DBP with lower affinity for 25-hydroxyvitamin D. If you have a protein with a lower affinity for 25-hydroxyvitamin D, you will have less of the bond form and you will have more of the free form. If you have more of the free form, you will have more 25-hydroxyvitamin D in the cell, and you will have more of the biological form of vitamin D. So more activity. Again, if you measure total 25-hydroxyvitamin D, you may find a lower concentration. If you measure free 25-hydroxyvitamin D, you may find a higher concentration or a different vitamin D deficiency status. This difference in affinity is still under debate, but let's say that the principle is there, and if that is true, then free vitamin D again is very important in multi-ethnic cohorts, or if you are measuring vitamin D, for example, in African Americans or in other ethnic groups. Just to summarize this part, as I explained you, the biological activity of vitamin D is directly linked to the concentration of free 25-hydroxyvitamin D due to the transport mechanism of free diffusion and due to the fact that 25 is then converted into the active form. In a healthy monoethnic population, the measurement of total vitamin D is enough, and it is representative of the free vitamin D concentration. So in that case, you should not measure free vitamin D. It will give you the same information as total vitamin D. However, in conditions with altered or modified DBP concentrations, or when you are evaluating different ethnicities, in that case, free 25-hydroxyvitamin D has to be measured. You cannot rely anymore on total vitamin D measurement. Let me show you a few key studies just to exemplify what I just discussed. And the first study is a study from 1996 from the group of Dan Bickley in San Francisco. They have studied two groups of patients. The first group is patients with liver disease, so cirrhosis patients. The second group was a control group with healthy people. In those two groups, they've been measuring the concentrations of DBP, albumin, total vitamin D, and free vitamin D. If you look at the data, DBP is lower in the liver disease group, as well as albumin. Total vitamin D is also lower in the liver disease group, about 50% of the concentration. But free vitamin D has almost the same concentration. What does it mean? Well, it means that if you look at total 25-hydroxy vitamin D, you will think that the group with liver disease is much more deficient and probably needs much more supplementation to reach optimal level. If you look at the concentration of free vitamin D, the concentrations are similar, even a little bit higher for the liver disease group. So which one is right? Should we look at total vitamin D or free vitamin D? Well, they linked it to clinical outcomes, and in that case, they looked at the prevalence of osteomalacia, which is a disease linked to vitamin D deficiency. What they found is that the prevalence of this disease was the same in the two groups. So they concluded that in that case, free vitamin D concentration is a much better representation of the clinical status of the patients than total vitamin D. Because if total vitamin D was really the correct marker, you would expect much more osteomalacia in the group of liver disease patients, which is not the case. So that's a very nice example and first example of measurement of free vitamin D in a specific disease, in this case, cirrhosis or liver disease patients. The second example is about pregnancy. As I explained you, DBP concentration is increased during pregnancy. And this is a study from a hospital in Berlin, a laboratory in Berlin. They took about 500 apparently healthy pregnant women, and they measured total 25-hydroxy vitamin D, and they measured free 25-hydroxy vitamin D in those pregnant women. And they made correlations between these two markers and other markers of pregnancy or of vitamin D, and calculated for all of them the correlation coefficient, the p-value. And you can see, for example, that for gestational age, there is no correlation to total vitamin D and a strong correlation to free vitamin D. For calcium, it is the same, p-value of about 0.24 for total vitamin D, 0.006 for measured free vitamin D. Bone-specific alkaline phosphatase, the same situation. Albumin, the same situation. Low-density lipoprotein, the same situation. Urea, the same situation. Adiponectin, the same situation, etc., 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 etc. What does it mean? It means that in a pregnant population, the measurement of total 25-hydroxy vitamin D is not anymore a good marker of vitamin D metabolism. In this population, there is a much better correlation of free 25-hydroxy vitamin D with a range of biomarkers. The conclusion of this study was really clear that during pregnancy, people should not measure total vitamin D anymore, but should measure free 25-hydroxy vitamin D. And I know this laboratory is starting now to measure free 25-hydroxy vitamin D in routine in the laboratory. The third study talks about the difference between vitamin D2 and D3, and this study was done in mice. And there has been some debate in the literature about should we supplement people with vitamin D2 or vitamin D3, and most of the papers were showing that vitamin D3 seems to be a little bit more efficient than vitamin D2. How was it established? Well, they looked at total vitamin D concentrations again. If you look at the concentration of free vitamin D, the situation is completely different. So what they did in this study, they took mice, they divided them into two groups. One group was supplemented with vitamin D2 only, and the other group was supplemented with vitamin D3 only. They then measure different parameters, and if you look on the graph, in the graph A, this is total vitamin D concentration. You hardly see a difference. There is no difference between the D2 and the D3 group. The concentration which are reached are the same for the two groups. Now if you look at graph D, which is free vitamin D concentration, you can easily see that the group which was supplemented with D2 has a much higher concentration than the group which was supplemented with D3. And the reason for that is the affinity of both forms for DBP. 25-hydroxy vitamin D2 has a lower affinity with DBP than 25-hydroxy vitamin D3. So if you supplement mice with vitamin D2, the increase in 25-hydroxy vitamin D will be much higher than if you supplement this mouse with vitamin D3. And as we have seen that the free vitamin D concentrations are very important, that gives another view or another idea of which one is more efficient between vitamin D2 and D3. In this case, vitamin D2 seems to be more efficient to reach higher level of 25-hydroxy vitamin D. This still has to be proven in humans, but this is an ongoing research, I know, by different groups. The last part of my presentation is about evaluation or measurement of 25-hydroxy vitamin D, of the free fraction. You know there are a lot of methods to measure total 25-hydroxy vitamin D. You have chemiluminescence assays, ELISA assays, LCMS methods, still a few RAAs, and then coming also a few point-of-care testings and some clinical chemistry assays as well. But how do you measure the free fraction, the free 25-hydroxy vitamin D? The very first method that was developed in the 80s is based on separation methods. So what you do in the first step is to separate the free fraction from the bound fractions. How do you separate the free fraction from the bound fraction? You can use two techniques, centrifugal ultrafiltration or dialysis. Then in another step, you take the free fraction which has been separated and you assay it with sensitive radioimmunoassay or with a very sensitive LCMS methods. This method is working very well and gives very accurate results, but it's not in use anymore because of different problems. The first problem is it is very, very difficult to reproduce, and the people who have been using this method in the 80s are not able to reproduce it anymore. It is highly time and resources consuming, so clearly not usable in IVD laboratories and not even in research laboratories anymore. It requires high volume of samples. We are talking about milliliters. It uses tritium and carbon-14 if you use radioimmunoassay, or very, very sensitive mass spectrometry techniques. And finally, it uses a few assumptions, so it's not a direct measurement. So this method was nice in the 80s, gave some good results in papers, but cannot be used anymore today. So what people have been doing during the last 10 years, they've been using calculations. You can also calculate free testosterone, for example. In this case, you can also calculate free vitamin D. And to calculate free vitamin D, you need this equation, on which you can see that you will need the concentration of total vitamin D, the concentration of albumin, and the concentration of DBP. So you need to do three measurements. From these three measurements and using the appropriate binding coefficients of both albumin and DBP, you will have the concentration of free 25-hydroxy vitamin D. By using a similar equation, you can also calculate bioavailable 25-hydroxy vitamin D, which is the sum of free plus the albumin bond fraction. But this bioavailable term is not really used anymore in research. What are the drawbacks of calculations? Well, the first one is very obvious. You need to do 3 assays per sample. Total 25-hydroxy vitamin D, DBP, and albumin. Ideally, you should even do a 4th assay, which is the genotyping of DBP. The reason for that is you are using binding coefficient in your equation, If you want to use the right binding coefficient, you need to know which form of DBP you have in your sample. As we have seen, there are 6 different forms. Each of these forms seems to have a different affinity constant. So if you do DBP genotyping, you will have the major form of your sample. You can use the right binding coefficient. You can have more accurate calculated results. The last problem, and it's really a big problem in most of the papers published in the last 5 years, is that DBP assays are not all of equal quality. There are 2 different types of assays. Some are based on monoclonal antibodies and some are based on polyclonal antibodies. And in this case, actually, the assays that are based on monoclonal antibodies are inaccurate. And the reason for that is that those monoclonal antibodies are actually too specific and they do not recognize all the forms of DBP. So in some samples, they will give good results. In some samples having other forms of DBP, it will give lowered results, which are inaccurate. Of course, if you have an inaccurate DBP concentration, you will have an inaccurate calculation of free 25-hydroxyvitamin D. So this is a big issue because some of these papers gave some conclusions in the last years. Most probably some of these conclusions are not correct, and those studies need to be repeated with another method of free vitamin D measurement. And now we come to the third method that was published very recently, and this is a direct measurement by ELISA. The principle of this method is described here. If you add a sample into an ELISA plate coated with an anti-25-hydroxyvitamin D antibody, you will have an equilibrium between the bound form, the free form, and the form that is captured by the antibody. If you play on the equilibrium between the bound form and the free form, you allow the free form to be captured by the antibody. Then there is a washing step that will remove the free form, which is not bound, and the bound form, and will only remain on the antibody a representation of the concentration of the free form. The rest is a classical ELISA, and it allows you to do a direct measurement of free 25-hydroxyvitamin D. People showed that this method is more accurate than calculations, especially in relation to PTH and calcium, which are 2 very important bone markers together with vitamin D. It only requires 1 assay instead of 3 or 4, and because it's an ELISA, it's accessible to any laboratory. Just to show you some characteristics of this assay, the measuring range is between 0.2 and 40 picograms per ml. As we have seen before, typical concentration for free vitamin D are between 2 and 8. So this is perfectly located in the measurement range. The total precision of the assay, or CV, for people which are familiar with this denomination, is about 5% or 4% depending on the concentration. The limit of blank and limit of detections are below the lower sample concentrations, which is fine, and the cross-reactivity of the assay towards 25-hydroxyvitamin D2 is about 80%. This method was validated against dialysis, and although the number of samples was not very high, it gave a good correlation and a good slope, which means that the method was correctly calibrated and validated against these dialysis reference methods. The method can also be automated for large cohorts and also for IVD use, and this is just an example of manual versus automated use. You can see on the left the standard curve, which are perfectly identical, and on the right the correlation performed on samples, which show that the assay as it is run manually or in an automated form are giving the same results. To conclude on this free vitamin D webinar, a few take-home messages. The first one is obviously that currently total 25-hydroxyvitamin D is used to evaluate vitamin D deficiency in all laboratories in the world. However, when DBP concentration or DBP form is different, total 25-hydroxyvitamin D is not a good marker anymore. In that case, free vitamin D concentration is really the key. Third point, if you do direct measurement of free vitamin D, it is much more reliable than calculations, and we have seen for what reason in the previous slides. Finally, I'd like to mention that with all the research that has been done, now the clinical utility of free vitamin D is really currently being demonstrated and implemented. So there are a number of laboratories and groups really working on the clinical utility of this new marker and really starting to implement it into their routine measurements. I would like to finish my presentation with this table, which again show in a different way the different cutoff values or the different reference ranges for vitamin D, depending on institution. In this case, Vitamin D Council, the Endocrine Society, the Food and Nutrition Board, and in some testing laboratories, other references. Again, this is a complex situation and people do not really know which one to take. And I hope, and this is my feeling, that free vitamin D measurement will help to harmonize, at least partially this table, and to provide new reference ranges which will be universal across diseases and healthy people. I would like to thank you very much for your attendance to this webinar. I hope you will consider free vitamin D as a new tool in your vitamin D research or in your research in general. I hope this was interesting for you, and I'm sure there will be questions, so now there will be a Q&A session. Thank you very much.
Thank you, Nicolas Heureux, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your questions into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, vitamin D is different from the other hormones in the sense that 25-OH vitamin D is measured. While the actual biologically active molecule is 1,25-OH2 vitamin D, does it make sense then to measure free 25-OH vitamin D, or is the free hormone hypothesis also valid for vitamin D?
Right. This is a very interesting question, and I discussed a little bit about that in my presentation. So you're right, vitamin D and 25-hydroxyvitamin D is not the active form of vitamin D. So is the free hormone hypothesis really valid for vitamin D, as it is for T3, T4, or TSH, for example? So as I explained, this is really linked to the transport mechanism of 25-hydroxyvitamin D inside of the cells. So 25-hydroxyvitamin D, to be converted into the active form, needs to be transported into cells. And this is done through two different mechanisms. And in kidneys, the main mechanism relies on total vitamin D concentrations. In other tissues, it relies on free vitamin D concentration. So in the case free vitamin D concentration is the most important. So in the case free vitamin D is transported by passive diffusion inside of the cells, the free hormone hypothesis is valid because the free hormone concentration in this case is directly linked to the concentration of the active form. So you're right, the situation is slightly different than for other hormones, but at the end the principle is still valid.
Thank you for that answer. Our next question is there an interest for measuring free 25-OH vitamin D in a pediatric population?
this is a question that is very often raised, because obviously vitamin D is very important in pediatric population. I would say yes and no. No, because in a pediatric population there is no change of the DBP concentration or no decrease or increase in DBP concentration. So in that case, you would think measurement of free vitamin D is of no interest in this population. Now the situation is different if you work on a pediatric population which has also some diseases. And I'm talking, for example, about kids having kidney disease. In that case, you have difference in the DBP concentration, and the measurement of free vitamin D is very important. The same if you have a multi-ethnic cohort of kids, of children. If you have blacks and white children in your population, again, you will have an interest of measuring free vitamin D instead of total. And this is the same situation for all population. If you look at elderly population or postmenopausal women or other population, you should always look at, do I have a DBP concentration which is increased or decreased by any condition, or do I have different ethnicities in my cohort or in my laboratory which require free vitamin D measurement?
Thank you. Our next question is, what is the cross-reactivity of the ELISA for 25-OH vitamin D2?
Thank you for that question. This is obviously an interesting question, especially in the United States where vitamin D2 supplements are used, especially for high doses supplements. The cross-reactivity of the assay for 25-hydroxy vitamin D2 is about 80%. And this is pretty difficult to measure actually, because you cannot do spiking experiments like you would do in other assays to measure cross-reactivity, because of the equilibrium in between the DBP present and all these things going around in the assay is not that simple. So what we did and what the people did was to take the same antibody, put it in a total vitamin D assay and measure the cross-reactivity in that total vitamin D assay. Because the cross-reactivity is really a property of the antibody. Whatever the assay you use, more or less, you will have the same cross-reactivity for one metabolite. So it's about 80%. This is not perfect, but it is, in my point of view, enough to use it at least as a tool to measure free vitamin D concentrations.
Thank you for that answer. Our next question is, how is the ELISA validated?
Very good question, especially when you have a new assay coming up in the field. People are asking, how do you validate it? What were the experiments performed, etc. So the ELISA was validated by different means. A direct validation against dialysis, as I showed you. So dialysis is obviously a reference method for free hormone measurements, not only for vitamin D, but also, for example, for free testosterone. So this was the first validation, and also a validation of the calibration of the assay. The other validation is slightly different. If you remember from the presentation when I presented that study on cirrhosis patients that was made in the 80s, this group has been repeating the same study with the same population. So the samples were not exactly the same, but the population was the same in terms of disease, in terms of age, in terms of different parameters. And they found the same concentration, the same mean concentration, using the direct measurement by ELISA and using at that time the ultrafiltration technique. So somehow this is an indirect validation, because you can have the same concentrations using this ultrafiltration and using the ELISA somewhere 25, 30 years later in the same population. So you have direct validation and you have indirect validation.
Thank you for that answer. Our next question is, are there published reference ranges for free 25-OH vitamin D?
Thank you for this very interesting question, which is obviously very important if you want to use this marker in IVD laboratories. You simply have to know from which concentration your patient will be deficient, sufficient, or insufficient in free 25-hydroxy vitamin D. There is no solid or concrete reference ranges published. There are a number of papers which have observed values in different populations, and there is a big paper which is about to be published very soon with this kind of ranges in a lot of different populations. This paper is a very good paper. I haven't seen it yet, but I know who is working on that. There will be a range of key opinion leaders which have provided free 25-hydroxy vitamin D data in different populations, and the leading group of this consortium is establishing statistics, mean concentrations, etc. from all these kind of different populations. So this will be published very soon and will really help the laboratories from a research perspective, but also from an IV perspective to establish their reference ranges for free 25-hydroxy vitamin D.
Okay, it looks like we have time for one more question. Our next question is the ELISA assay you cited IVD marked CE or FDA?
This is obviously a very important question for those of you who are working in clinical labs, in IVD labs. At the moment, I know the ELISA assay is only for research use only. But as far as I know, end of this month, or actually beginning of this month, it will be CE marked, so it will be usable at least in Europe as an IVD assay, also in countries which accept the CE marking. For the FDA marking, I'm afraid we should wait a little bit more, and one of the reasons for that is that there is no predicate device for free vitamin D in the US. So the way the FDA works requires an equivalent assay for a new assay to be accepted by the FDA. As this free vitamin D ELISA is a new assay, there are a lot of discussions with the FDA how to proceed, and it will take a little bit more time for the US people and US laboratories to be able to use this ELISA.
I would like to once again thank Nicolas Heureux for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February of 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.